Hey everyone, my name is Jordan Rudeen, and this past summer I had the awesome opportunity to go to Costa Rica and study the geographic comparison of bioacoustics between disjunct populations of Dusky Nightjar, also called Antrostomus saturatus. To start, let's talk about this cute little fella we have right here. This is a Dusky Nightjar, and is part of the family Capermolgidae, which is the same family that the common nighthawks we have here in Idaho are in. Another name for this group is actually called the goatsuckers, and in fact, the Latin name for goatsucker is actually Capromulgus. This family name originated from ancient people thinking that these kinds of birds would actually go out at night and suck the milk from goats. Curious, isn't it? You can actually find Capromulgids on every continent except for Antarctica. However, despite the large distribution, this family remains a relatively unknown and understudied group of birds, even though in fact, they're actually very cool. Dusky nightjars are part of the Neotropical order of nightjars, and they are endemic to Costa Rica and small parts of Panama, as you can see on this map. There are 98 species of the Capromolgidae family worldwide, and in Costa Rica there are about 12 different species, including the dusky nightjar. Dusky nightjars can be found in the subalpine paramo shrub areas of the Costa Rican mountains. They are crepuscular, which means they become active during dawn and dusk, which is also when they feed. Dusky nightjars are aerial insectivores, meaning they catch insects on the wing. They feed by sitting on a low branch or a fence post like the one in this photo, and when an insect flies by, they will fly up, snag it, and return back to their perch. Dusky nightjar prefer this ambush style of hunting as opposed to their cousins, the nighthawks, who fly around in the air and outmaneuver their prey. Before I move on, I'd like to talk about an important concept that characterizes the tropics. In 1967, Dan Jansen wrote a paper called Why Mountain Passes Are Higher in the Tropics. In this paper, he talks about a phenomenon that distinguishes temperate latitudes from those in the tropics. He states that the thermal zones in temperate latitudes are more dynamic than the ones in the tropics. This is due to the reduced seasonality in the tropical latitudes. As you can see in this diagram I made, there is a larger change in the seasonal temperature on a temperate mountain than on a tropical one where there is nearly no change. In response to this, species in temperate latitudes have a larger tolerance to temperature, and thus a higher ability to adapt to changing landscapes that occur from temperature changes. On the flip side, tropical species do not have as large of a tolerance, so they are more specialized in habitat type. As the climate changes and average temperatures rise in the tropical mountains, species are forced up the mountainside and further away from other subpopulations. This growing isolation can lead to allopatric speciation. And in case you don't know, allopatric speciation is speciation due to geographic isolation over time, and this was a point of interest for our study. So, why the dusky nightjar? Well, for one, they are listed to reside at about 1,500 to 3,100 meters in elevation, which actually subjects them to Jansen's theory. Along with this, they are already known to be disjunct, separated into 3 distinct subpopulations across Costa Rica. On this map here, you can see the separation between the mountain ranges. In addition, little is actually known about these cryptic little birds and their fellow neotropical cousins, like potus, oilbirds, and frogmouths. Any information we could gather would be beneficial and serve as useful steps towards better understanding the family Capromolgidae as a whole. And finally, dusky nightjars are a good model to use for the future study of high elevation and range-limited species. There are many other animals affected by the same isolation as the dusky nightjars, so gaining knowledge on these birds will inherently boost our ability to study other related species. We chose to use bioacoustics for a couple of reasons. First, they're a great starting point for a pilot study like ours. This technique is relatively cheap, easy to collect data with, and non-intrusive to the animals you want to study. This could also help in a case where the ability to capture a species is hindered. Also, bioacoustics are additive in their nature. Combining them with DNA, morphometrics, or mark and recapture techniques can greatly enhance the biological study of an organism. When it came to our study, our goal was to describe and compare the bioacoustics between all 3 isolated populations of dusky nightjar. And our hypothesis going into it was that despite the disjunct status of the populations, there will be no evidence of speciation between them. My reasoning for this hypothesis is that when I listened to the pre-recorded audio of the birds before the trip, and then I heard them in person, I could tell no audible difference between the two. Fieldwork consisted of nocturnal surveys conducted at dusk at 4 different locations on 3 mountain ranges. We surveyed at Monteverde Cloud Forest Reserve in the Tilarans, Volcan Poas and Turrialba in the Central Range, and finally at Cerro de la Muerte in the Talamancas. We drove along mountain roads and would stop every 100 or so meters to get out and do audio playbacks, using other Dusky Nightjar calls that we have from the app Merlin. To do this, we used a portable speaker and would play for a minute or until a bird started singing. When one did sing, all lights and playbacks would be turned off and the person with the audio equipment would then start recording. We'd be listening for something like this. Back in Idaho, we then began to compile and analyze the data we collected in the field. We decided to use the popular audio analysis program Raven to do so. Once in Raven, we chose 8 song characteristics to be measured and collected as the variables for our statistical tests. They were song length, inner song length, song units per minute, frequency 95, maximum frequency, peak frequency contour, high frequency, and low frequency, with frequency 95, maximum frequency, and peak frequency contour as being our representatives of dominant frequency. But to do this, we needed to define what a song was. The differentiation between a song and a call is still debated amongst ornithologists, so there is still room for interpretation here. So we defined it functionally as something that serves a purpose and is comprised of multiple notes. The Dusky Nightjar has a very short and repetitive song, as seen in the spectrogram, which was useful when analyzing it. We used a student t-test in Excel to test the significance of the results once we were done. These next few slides include box plots that represent the comparison between the variables at each mountain range. The p-values from our t-tests are displayed at the bottom of each group. For song length and intersong length, the units of measurement were seconds, and they were pretty straightforward to measure. Units per minute just takes into account the number of songs a bird repeats in a minute. Now you may notice that the p-value of this variable is green. This is because its t-test produced a value that was below 0.05 and therefore statistically significant. Moving on to the dominant frequency variables, they were measured in hertz. We chose these variables because we were not sure which one would give the most accurate reading, so we took all of them. Our last few variables were high and low frequency and were also measured in hertz. These variables are a nice way to round off our description of the songs. As you could probably tell from the results, we did not get any data from the Tilarán mountain range. Since this was our first site, we were unsure about the success of the project. In fact, this led us to create the motto, "You can't say we didn't try," or YCSWDT. We searched for singing nightjars for 8 days and over 20 hours at the Monteverde site in the Tilarans, but never heard a single nightjar song. We tried both evenings and mornings, but had no success during either time. Our best guess at this point is that, given their propensity to sing, it was a combination of the rarity of the Dusky Nightjar in that region and the lower elevation we surveyed at that led us to our lack of data. This being said, we did not find a significant statistical difference between the Central and Talamanca mountain ranges. Our results fail to reject our hypothesis and do not show evidence for a speciation event due to allopatric speciation. However, more evidence apart from bioacoustics will be needed to confirm this. Now, as I mentioned earlier, the song units per minute variable did show a significant difference between the populations. Literature on related capomulgids do show that song tempo can vary based on the arousal of the bird. We suspect that this variance could be due to a difference in the breeding cycle of each population. And lastly, while recording, we observed that the birds displayed aggressive behavior towards the speaker and would on occasion fly right at it, missing our faces by inches at times. It was this behavior that got us thinking that capture using mist nets would be a viable tactic to use in a future study. By capturing Dusky Nightjars, DNA samples could be taken and GPS trackers attached to the birds. These two variables would allow for a more comprehensive study of the biology of these species and could confirm or deny their isolated status. Additionally, I would love to get audio samples from the breeding season since we were there shortly after this occurred and the birds' calls may be different at that time. In conclusion, the Dusky Nightjar is a good study system for understanding both the speciation events of the Costa Rican mountain ranges as well as the effects of climate change on range-limited and high-elevation species. To wrap things up, I want to reiterate that biogeography is an important and growing phenomenon. As humanity continues to grow and encroach upon the natural world, it has become imperative that we find ways to live in harmony with God's creation. As technology improves, bioacoustics is becoming a great option for researchers to study species like the Dusky Nightjar and promote tropical biodiversity at the same time. But it is important that we scientists always remember there is more work to be done. I'd like to thank God first for this amazing opportunity to study and enjoy His creation. I'd also like to thank Monteverde Cloud Forest Reserve and Quirk for letting us use their facilities and trails. I'd like to thank August Thornton and Carly Gilmore for their various help in whatever I needed, the ever so illustrious Dr. Cassell for his expertise in bioacoustics, and of course my rockstar team, Professor David Hilley and Elise Griffith, who not only were a huge help, but have become dear friends of mine. Thank you so much for listening, and please stay safe, everybody. Pura vida.